Go and get started. Okay, so I've written up here the reading for today's lecture and for Wednesday's lecture. We probably in fact won't get to this today, but just go ahead and read those couple pages, and then this is what we'll do on Wednesday. So I'm not going to have office hours today or on Friday, but I'll have instead office hours is listed here on Tuesday and Wednesday. Okay, and then of course there's a review session Wednesday night that you all know about. Okay, so we left off, we earlier in the series of lectures here, we talked about the process of DNA replication and actually talked quite a lot about that. And then before the break, we talked about the processes of transcription and translation, which allow you to take information from DNA then and convert it to RNA and then finally to a protein sequence. And so we went into detail about that. And the main players in this kind of process, so for DNA to DNA replication is DNA polymerase, and then RNA polymerase for DNA RNA. And then the ribosome is the main player in reading the RNA sequence and then turning that into protein along with the tRNAs. Just to be complete, this is usually the way that information transfer is drawn, and it's sort of one of the central dogmas. But in fact, there are a few exceptions to this I thought I'd just point out. So there are some viruses that have just RNA for their genomes, and they actually never turn into DNA, so they have an RNA-dependent RNA polymerase, so they can make RNA out of RNA. So an example is poliovirus. So in fact, its RNA genome can be translated straight into a series of proteins, and it can also replicate itself as an RNA. And then there are a few cases, too, where viruses have RNA, and they can actually reverse transcribe themselves into DNA. So that's sort of a rare exception, but HIV virus, for example, is able to do that. It's an RNA virus, and once it enters the cell, it actually has a RNA-dependent DNA polymerase, so it can turn its RNA into a DNA sequence and integrate itself. Okay? But for most of what we have talked about in the last lecture, we focused in this direction and the processes of transcription and translation. And we talked about how transcription is a process that occurs, at least in eukaryotic cells, in the nucleus, and then the mRNA is processed, sent out into the cytoplasm, and then translation occurs in the cytoplasm. In the case of cells without a nucleus, transcription and translation can be coupled together, essentially occurring one right after the other on the same strand of RNA being made. Okay, so, and then just very quickly to review. So we talked about RNA polymerase as the enzyme. It's actually an enzyme that's composed of multiple subunits of protein that's able then to use the DNA as a template and then create an RNA strand off of that. And then the idea that you could then read this RNA in triplets called codons, and then the ribosomes would help you turn that into an amino acid sequence. And we went through a little bit about how that works, where the RNA is feeding through the ribosome, and then there's a site where the tRNAs can come in, which have anticodons that match those triplets on that mRNA, and then that forms a linkage of polypeptides, and so you get the corresponding amino acid sequence. Okay, and there's 20 amino acids, so there's some redundancy in the code that you can have then. And then this is just that table showing you if you have the RNA sequence, then you know what amino acids are going to be encoded. A couple unusual ones. So, for example, you always start with an AUG, a methionine, right? And then you've got 3 stop codons. And you can see that some amino acids can only be encoded in a single way, and others, for example, leucine here, can be encoded by 6 different codons. Okay, and the key, in addition to the ribosomes, the other key are the tRNAs, which have the anticodon sequence on them, and they're coupled to the appropriate amino acid. And so they enter the ribosome, and then you get the reaction that couples together the amino acids, and you get the forming polypeptide chain. Just as an aside, there's not necessarily a tRNA with every possible codon, or anticodon, sorry. So you're probably, like if you do exercises in the book, you're taught that you can write an anticodon for every triplet, but in fact that doesn't always occur in every type of organism. So sometimes you can actually use a tRNA and go ahead and use it for 2 different codons. So for example, histidine is either CAC or CAU in the mRNA sequence. And so these would be the 2 anticodons, but in fact you can have cells that just have a single anticodon. And what'll happen is that can actually pair up with either of those. And that you'll see is consistent with the genetic code, and it still gives you the right amino acid sequence. Okay. So that's where we left off last time. So we're going to continue now, and we'll start talking about mutations. So we talked about you have a DNA sequence, you take that RNA and protein. So what really is the effect of mutations? And we'll start by just focusing on mutations that are actually in the part of the gene that encodes the protein, okay, the coding region of the gene. So mutations are changes in the genetic material, and point mutants are chemical changes in just one base pair of the gene. And you can also then have deletions or insertions. You can have a few or a lot of nucleotides inserted or deleted in addition. Okay, and in fact, because of the way the codon system works, a single nucleotide change can lead to the production of an abnormal protein. Okay, and here's just a very simple example from hemoglobin. And so this codon here is encoding in glutamic acid, but if this nucleotide is mutated, okay, so instead of being a CTT here, it's a CAT, then you're going to get a valine instead, okay? And this actually is the mutation that causes sickle cell anemia, so that hemoglobin molecule no longer behaves normally and can't actually bind oxygen in the normal way. So that's an example where a single base pair nucleotide change will give rise to a mutant phenotype, okay? And you can get, so we have different names for the different kinds of changes that occur. So we call it a substitution when one nucleotide is replaced by another. And then you have silent mutations. So those are changes in a nucleotide that don't affect the amino acid produced by the codon, and that's because of redundancy in the code. All right. And those are called silent mutations. We'll come back to that in a minute, though. Okay, missense mutations are just like the example I showed you before, where they still code for an amino acid after the substitution, but it's not the correct amino acid. It's not the amino acid that was there before the mutation. Okay. And then nonsense mutations are ones that change the amino acid into a stop codon, and those nearly always lead to a non-functional protein. So if they terminate the protein early, then that's going to be non-functional. If the stop codon is introduced right near the normal stop, occasionally that's still an okay protein. Okay. But generally, nonsense mutations in the coding region will give you a non-functional protein. Okay. And then, like I said, you can also get insertions and deletions. So instead of just a single base pair changing, you can get several nucleotides either gained or lost. And that can either be a single nucleotide gained or lost, or actually thousands or even a million base pairs of DNA when strange things happen that get inserted or deleted. Okay. And these generally have pretty serious effects when they're in the coding region. Okay. And one of the ways that they can cause a serious effect is to cause a frame shift. So if you have a triplet pattern that you're looking at, you insert a single nucleotide, then everything after that nucleotide is going to be shifted in frame. Okay. And usually what happens then is you run into a stop codon pretty quickly, and then you have the wrong amino acids and you terminate early as well. And those are called frame shifts. Okay. So here are just some examples of this kind of thing. So again, a normal sequence, and then if we introduce this extra AT base pair, then we introduce a stop right away, and so then that would terminate. You wouldn't get the normal polypeptide chain. Okay. Okay, and then the same thing would happen. Example, you would have a deletion here, okay, and then what would end up happening is that you would frame shift and you would start changing amino acids after that point. Okay, so those are all examples of the kinds of changes that you can get. Okay, so how do you get these kinds of mutations? Okay, well, one way, of course, is that we talked about you have occasional errors in DNA replication, so those occur sometimes. But the other thing that can happen is that you can have mutagens. So you can have chemicals or physical agents that cause mutations that damage the DNA. So there are a lot of chemical compounds which are actually highly reactive, and so they can end up mutating the DNA sequence. Okay? And so if that mutation occurs, in a large intervening region between genes or inside the intron of a gene, there's a chance that that won't have a phenotype. Okay? It varies. It depends exactly on the function of that. If it's in the coding region and it actually, for example, inserts a nucleotide, causes a frame shift, or things like that, it could quite well have a phenotype then. Okay? So back to this silent mutation. So you're told that silent mutations have no effect on the amino acid. Okay? And so most people then assume that there is no phenotype from that. Okay? So why up here would we say that there'd be no phenotype from that? Anyone? If you change the codon and it's still encoded the same amino acid in the coding region. Yeah, so you expect that not to create any problem because it should actually make the exact same protein. Okay, but the reality is that every now and then this isn't true, and there are a couple examples now where we can see that what we call a silent mutation actually has a phenotype, and it has to do with the prevalence of different codon usage. Okay, so this table here, okay, is a different kind of table. It's a codon usage table. It shows you how frequently in a genome, okay, a particular codon is used for an amino acid. So, for example, if you look at methionine, it says 1, and that's because there's only one codon for methionine, so that's what you always have to use. Okay, but if you look at some of the others, so you can see that some are fairly evenly distributed. So glycine, you have 4 possibilities, and they're roughly a quarter of the time each one, right? But in a few cases, so if you look here at leucine, there's 6 different ways to encode it, but you'll see that this one is used 40% of the time. And these two, the CUA and the UUA, are very rare, right? They're 0.08 and 0.07 respectively. So they're rare codons. They usually reflect the fact that those tRNAs are not very abundant. So there's selection not to use those codons. So it turns out that if you have lots of those rare codons, or a rare codon in a particular location in a sequence, it can actually cause a problem. And here's an interesting experiment that was done. So alcohol dehydrogenase is an enzyme that helps you break down ethanol. And in this case, experiments were done in flies and Drosophila. So they have a single copy of this gene. And it has 10 leucines in it. Okay, and experimentally, if you make all those leucines CUA, so that's the rarest codon for leucine, okay, and you put that gene back in, so you'd assume beforehand that, well, that shouldn't cause a problem. It's going to encode exactly the same protein. But it does. You can see that this is what's called an LD50, how much ethanol it takes to kill half the flies. And so you can see if you replace all 10 leucines with this rare codon, you have a significant drop in tolerance to ethanol. Okay? And it turns out the reason for this is that actually if you try to incorporate all these rare codons, it slows down the whole process of making the protein. Because ribosome has to wait longer for a tRNA to show up. So everything slows down, and you actually end up making less alcohol dehydrogenase in the cell. So it lowers the protein level, and then it makes the cell more sensitive to ethanol, the animal more sensitive to ethanol. Okay? So that can happen. So that's this example. Another way that it can happen is that it actually causes the chain to polymerize, the amino acid sequence to be made slightly slower. And proteins have to fold as they're being made. And it turns out that if you make the protein at a slightly different rate, sometimes it folds in a slightly different way, okay, because of other interactions that occur as it's being made. And so sometimes, for example, this is a multiple drug resistance gene, and there's a silent mutation in it that has a clear effect, and it's because it incorporates a rare tRNA or a rare codon. It slows down the rate that it's actually making the protein, and then it folds differently, and it doesn't function properly. Okay, so sometimes those silent mutations are detectable. Okay, so now we're going to go ahead and move on to trying to understand how you regulate where genes are expressed, when and where. And this is a really important thing, because in all the cells of your body, okay, almost all the cells of your body, there's a few little exceptions, but pretty much for all the cells in your body, every cell has the same DNA in it, right? It has the same complement of genes. So why aren't all your cells exactly the same? And the reason is because the genes are not being, well, the bottom line is it's because there are different proteins and different biochemical activities in different cells, okay? So for example, you have hemoglobin in blood cells, and you don't have that in these other cells. You have cells that secrete enzymes for breaking down food from your stomach. You have cells that are specialized for absorbing food in the small intestine. You have channels that are useful for sending electrical signals in neurons and so on. And those are specific to those different kinds of cells, okay? And there are many ways to generate specific proteins in each cell, but one of the primary ways of doing it is transcriptional regulation, to regulate which genes are actually transcribed when and in which cell, okay? So for example, the hemoglobin gene is only transcribed in developing blood cells and in no other cell types in your body, okay? Another example is your muscle. So there's lots of proteins that are very abundantly expressed in your muscle that are only supposed to be in your muscle. So it doesn't do you very well if your cells in your brain start expressing proteins that are normally supposed to be in muscle. That's not what they need, right? So how do you ensure that, okay? And this just sort of illustrates, again, some of the proteins that are there. And what happens is that there are many ways, again, to regulate that. We'll briefly tell you a few, but today we're just going to focus on that process of transcriptional regulation, making sure that those genes you need are only transcribed in the cells that should have them, okay? And then this is sort of a central property then for any organism that's multicellular. When you have lots of different cells that are doing different things, you need to ensure that the different cells have the right proteins and they're actually expressing, as we say, the right gene, okay? And like I said, there are many ways to do it. Some ways have nothing to do with regulation of transcription. So for example, you can have proteins that are actually expressed in every cell of your body, but they're only functional, say, when they're phosphorylated, and that phosphorylation may only occur in a few cells that need that active protein. So that would be one way to regulate something like that. You can also have the same gene transcribed in 2 different cells, but they're spliced differently so that you get different protein products, okay? And you can also have a protein that's made, but it has to be cleaved into an active form, and that cleavage only occurs in certain parts of your body and not others. So you would have different biochemical activities, but it would have nothing to do with transcriptional regulation. But it turns out that transcriptional regulation is probably sort of the key mechanism, though, for controlling this, right? Despite the fact that there are many other mechanisms, transcription is the main one, okay? So both prokaryotes and eukaryotes alter gene expression in response to a changing environment. Okay, what genes you are turning on and turning off in different cells has a lot to do with the environment that those cells are in. In an organism like you, that environment is very controlled. So having that control of the environment and the interaction of cells as you were an embryo developing is how you got all the right cells in the right place. Okay, so that in multicellular eukaryotes like ourselves, that pattern of gene regulation is very important in the process of development and getting pattern to occur properly and getting a baby to be born that has the right organs in the right place and so on. Okay, but it turns out though that bacteria, even though they're just single cells, right, they also have to have a system of gene regulation. They don't want all their genes on all the time. Okay, and the reason is because that's a waste of energy. The main reason. Okay, so you can imagine that you have, say, 10,000 different enzymes, right? And it can do lots of different things. And some of those you need all the time. So you need RNA polymerase, for example, all the time. So it's always being expressed, okay? But there are other gene products that you only need at certain times. And what we're going to focus on for a minute now is gene products that are involved in biosynthetic pathways, okay, and making certain molecules, okay? And in bacteria, they're controlled in an interesting way. They're controlled by what are called operons. So if you have a series of enzymes that are required to make something, those enzymes essentially are linked together. They're actually made as a single messenger RNA, and there's stop and start codons in that messenger RNA that can be used to make, say, 3 or 4 different proteins simultaneously. And so an example of this is what's called the trip operon. And so this is used to synthesize tryptophan. Okay? And so there are these 5 genes that are involved in making tryptophan from this precursor, charismatic acid. Okay? So the thing is, though, that bacteria often find themselves in an environment where there's plenty of tryptophan in the environment. And of course, tryptophan is essential. It's essential amino acid, right? They have to have it. So if the environment is full of tryptophan, then it's a waste of their energy to be making all of these enzymes. Okay? They don't need it because they could just absorb tryptophan from the environment. But if tryptophan runs out or runs low, then they need these enzymes in order to then go ahead and synthesize tryptophan from other precursor molecules that they may have access to. Okay? So they regulate this process so that they're not always expressing these enzymes. Okay? And so, like I said, the enzymes here are encoded in what we call an operon. So there's a single gene, DNA, that encodes all 5 of these genes in a row. Okay? And it's transcribed as a single mRNA, but then there's position, stop, and start codon so that it can make all these different peptides, okay? All these different proteins. So it's very rare in eukaryotes like ourselves to have any setup like this where you have multiple proteins coming from a single mRNA like this, but it's very common in bacteria. And of course, it's a very nice setup because what it means is that when you need those enzymes, you can make all 5 at once in a very concerted way. Okay? And in fact, you'll get equal molar ratio of them and so on. All right, but you only need them when you only want them to be made when tryptophan is not around. Okay? And the way the system works is a repressor protein that's encoded by a different gene called TRPR. So it makes this protein. Okay? And if there's normally, if there's no tryptophan around, it's in an inactive state, and the promoter binds here in front of this operon, and it reads this mRNA and it makes these enzymes. Okay? So that's what happens when there's no tryptophan around. Okay? But if tryptophan enters the cell, okay, then the bacteria can make use of that. It doesn't need the enzymes to synthesize it, but the tryptophan can also bind to this protein here that was made from the TRPR gene. And then that protein complexes with tryptophan, and then it binds to what's called an operator sequence just in front of the promoter. And now polymerase can't get through. Okay? And so now that operon doesn't get transcribed, and those enzymes don't get made. Okay? So it's a very elegant system. So it allows, in a very simple way, the presence of tryptophan to shut off this gene. Okay? So it acts as a co-repressor. So tryptophan plus the TRPR product bind together, sit down in front of the promoter, and then stop RNA polymerase from being able to go through there. And then you don't have tryptophan. When the tryptophan runs out, then the tryptophan comes off the repressor, and then polymerase gets through, and you start making the enzyme again. All right? And then here's another system that's also described in your book that works in the opposite direction, but in a similar conceptual way for beta-galactosidase. So beta-galactosidase is the enzyme that can break down lactose into galactose and glucose. Okay? So if there's plenty of glucose around, and if there's no lactose around, there's no reason to make beta-galactosidase. And again, it's part of an operon, so this permease actually allows beta-galactosidase to come into the bacteria as well. Okay? So in this case, what happens is if that lactose is absent, okay, then this repressor actually stays bound to the operator, and the polymerase can't go through. And now it works sort of in the reverse way from the TRP one, that when lactose is around, it binds to the repressor, inactivates the repressor comes off its binding site on the DNA, and now polymerase can go through and make those enzymes. Okay? So it achieves the same sort of thing. It acts as a sensor for the presence of this molecule in the environment. And then the bacteria can respond by turning on and off the appropriate genes. All right? So that's a sort of simple way that it works. And the book shows you one more complication on this, that if you had lots of lactose around and glucose was also scarce, right, it turns out that what happens is that causes high levels of a molecule cyclic AMP, and there's another protein called CAP, which is actually helping RNA polymerase bind and get started. And the reason for this system is that you can imagine that if you actually have a situation where you have lactose, but you also have plenty of glucose, you don't actually need to waste your time making beta-galactosidase to convert the lactose and make glucose that way because you have plenty of glucose. So what happens in this case is that this CAP protein doesn't bind, and then in fact polymerase can't get bound, and this operon stays off. Okay, so it's a nice sort of balanced system. And in general, for bacterial systems, it's very compact. All what we call the regulatory part of the gene sits just around the promoter. So these are binding sites that can either get in the way of RNA polymerase getting through, or get RNA polymerase to bind on there and get started. Okay, and they do so right around that promoter, and then they get polymerase to make or not make that operon sequence. Okay, does everyone follow? All right, so that's a pretty simplified. That's how it works in bacteria, and it's a fairly straightforward system. Okay, but it turns out that in multicellular organisms, right, things get a lot more complicated. So again, now you have a situation where in the organism, right, the cells are more or less genetically identical. They have all the same genes, but you need differential gene expression. So what you have now is again a slightly more complicated system, and it's more spread out over the genome. So the control regions are not right there next to the promoter. For the most part, in multicellular organisms, the promoter is really just the place where RNA polymerase can bind. And what happens is that there are control elements that sit at various distances from the promoter that are actually going to get the gene to be transcribed or not transcribed from that location. Okay, and these control elements are also called enhancers. Okay, and they are the sites where transcription factors bind. So transcription factors are proteins that recognize and bind to specific DNA sequences. Okay, so those enhancers contain usually clusters of those sequences that those transcription factors can bind. And when the transcription factors bind, they either promote transcription at that gene or they inhibit transcription at that gene. Okay, so they're what we call them the regulatory elements. And these enhancers can be located very close to the promoter, or in some remarkable cases, they can be located a million base pairs away from the gene that they're actually regulating. In fact, they can be located several genes away from the gene they're regulating. Okay, and we'll talk about the reason that works is because they actually bend around and come to the gene that they're regulating. Right. So when we talk about a gene, particularly when we talk about it in a eukaryote, a multicellular eukaryote, it's not just the area that's being transcribed, okay, that encodes the protein that's important. So these regulatory elements, these enhancers, and they're distal and proximal, ones are close, ones are far, and so on. And they can sit upstream of the gene, they can sit downstream, they can even be in the introns. So these end up being very vital to controlling when and where a gene is expressed. And mutations in those can be just as bad as mutations in the coding region. So you can imagine if there's a single regulatory element that enhances transcription and you mutate that, that's also just going to shut off the gene. Okay, the protein coding region will be unchanged, but the regulation will get messed up. All right. So in reality, a gene is all of this kind of information, not just the coding region, but also all these regulatory elements. And people used to think, you probably have heard the term junk DNA, that there was lots of DNA, but because it didn't code for anything in your body, in fact, the vast majority of DNA in your cell doesn't actually encode a protein. People thought, well, you can't really be doing anything. It's sort of junk DNA. But starting probably,